Okay, let's do this thing. Okay, so let's go ahead and start on this middle side of the table. We'll go around like BS, then come back to the outside. Concentric circles or ovals. Is that a thing? Do we have concentric ovals? Okay, I don't geometry. Ready? First question.
I was wondering because I know in my previous other is one has two strings, each part on my each one would be a different type. So I don't know why the competition,
Okay, so you're looking for the probe that's going to have the strongest interaction. You're thinking about a probe for strongest interaction. What concepts I'm trying to get you to think of? What makes a probe interact either more strongly or less strongly with the target DNA? Yes.
I guess like length as well as in RNA or DNA.
Length, RNA versus DNA. In that question, the probe that is the correct answer, how long is it compared to the other probes there? Is there a difference in length?
The two longest ones are about the same.
Okay, so you can basically go through, but I know the longest things are going to probably be better, right? So I'm looking at the length, longer, stronger association, better hybridization. And then we're going to look for RNA versus DNA. RNA is going to tend to have a stronger hybridization with DNA as opposed to DNA with DNA. Does that help with that question? Okay, question answered. We need to make sure it's clear before we move on.
Wondering on why, there's no one that we know like why are these or it's just that, but I think.
You're going to have to know why to answer the questions.
Okay.
So remember, actually for this particular one, even just know that our RNA hybridizes better with DNA is partially because of the uracil interaction. That's really one of the core reasons why you don't need to know the deep mechanism, but this whole relationship of RNA hybrid greater than RNA with DNA is greater than DNA to DNA. And what you want to make sure you keep separate in your mind is the stability of hybrids versus stability of monomers or individual DNA strands or RNA strands. So if you think about, this is the hybrids. If you think about stability of monomers, it's going to be DNA is more stable than RNA. This is because the potential for this OH group to undergo hydrolysis in acidic conditions and at higher temperature. Okay, these two concepts are clear, yes? Okay, on to the next question. Oh, it's on the same topic?
Oh, we're going in.
Okay, so let's keep going around. Yes.
Why is it easier to
Okay, so let's write the question for everyone. We're starting off with a. Okay. So we're adding in the LU2 gene, correct? Okay, and equisol resistance. We're transforming it into this oxytrough. Okay, and then the question stem is asking you what media? What are our answer choices? You right there, us. Okay, wait, what's okay? Number one was without leucine or ampicillin, right? Number two, with leucine and no ampicillin. Three, no leucine with ampicillin. With ampicillin. And what is correct answer marked? Okay. So I'm guessing the ampicillin part is really clear and like really straightforward and easy to people. So we have to add the ampicillin because this now has resistance. So you can go ahead and be like, okay, anything with no ampicillin, it's not going to be the right answer. And then we're basically still trying to decide whether or not we need to add leucine. This one says no leucine. The reason why we don't need leucine is that we're adding in the LEU2 gene. So now it has the ability to synthesize its own leucine. So you're trying to think of genotype versus phenotype, and what does the genotype mean? Once we give it this LU2, it means that it can now make its own leucine. So it doesn't need leucine in order to grow. So is that clear? The process of like auxotroph, it can't synthesize something, it needs that one nutrient in order to live. Okay, yes.
So whenever it transports like the one without the loose, what do we need to add leucine because it doesn't have it? That's not really confusing.
Okay.
Whenever it transports.
So think of this as, this is a plasmid, this is the genetic material. When we do transformation, this genotype is taking up these new genes. So think of it as like LU delta plus, now I have this gene, and plus now I have ampicillin resistance.
So it's not a knockout.
So this was a knockout, doesn't have it, and now you're adding something to it.
Oh,
It's almost like saying 0 + 1 plus ampicillin. And now I have this. So this not having it doesn't matter, because now you have it now.
Okay, that's what I was,
Go ahead.
so when it has ampicillin resistant, it must be given ampicillin in the media to show that it is not growing out of transplant.
So what the ampicillin does, this is selection. So remember that we want to, whenever we transform, is make sure we can grow the things that we transformed, and everything else we want it to die. All yeast and bacteria, they're going to be sensitive to antibiotics. That's what these are, like ampicillin, kanamycin, things that end in sin. They are all antibiotics meant to kill off whatever's there on the plate. So by giving it resistance, it means that if we add in the ampicillin, we don't kill this particular thing that got transformed. Anything that didn't get transformed didn't get a resistance gene, and so it's going to die in the presence of ampicillin.
It's gonna grow.
So this will grow in the presence of ampicillin, exactly. Yes,
With the delta, you consider the oxygen.
yes. This is your oxytroph. Nice spell. Oxytroph.
And you're adding it to the oxytocin.
Yes,
Like, okay, okay.
we're starting off with the oxytroph, then we're transforming these genes into that oxytroph. Any other questions about this topic, this question? Okay, we're gonna go move on to the next one.
They're asking about practice exam questions here.
Yes, we're starting off with practice exam questions.
Not the going, I think, turn.
Yes, we're gonna go with this inner circle. So you sign the inner circle, so you're good. And then we're gonna go to the outer circle. Okay,
The question I was asking about having to being treated like extremely acidic solution. Why can't it be renewed? Would it be able to renew if we were to turn back to the, like,
Wouldn't that base?
yeah, so this is the concept I'm just gonna touch on. Just know, like, you won't have this concept directly on the exam because we didn't hit it in lecture. So since everyone didn't hear it, I want to make sure it's not in the exam. But when you acid hydrolyze a DNA backbone, it doesn't just denature. The actual like phosphodiester bonds in the backbone break. So you destroy the DNA in really, really extreme acidic conditions. So it's never going to be able to come back together because you just destroyed it.
Is
Oh.
that only for extremely acidic or extremely
For
basic?
acidic, extremely acidic.
So extremely basic, we can add.
Acid, and they'll come back together. Yeah.
So then how would you return extremely acidic solution back to normal? Wait.
I mean, you return it back to normal pH, but your DNA is destroyed.
Oh,
Like you can't rescue your DNA. There's some other carpet spaces you want to come further inside. That floor looks kind of cold, and I don't know.
What pH is extreme acidic, you would say, when do we start getting kids two and so on, is extreme acidic?
I mean, I would say that, but don't think of a hard number because it all depends on, there's other factors involved. So the idea is that I want you to grasp the concept. When we go extreme, it's bad. And depending on like situation, that number is going to shift.
And extremely basic doesn't break the back.
Doesn't break the backbone. Correct. Yes.
so I can renew it. Extremely basic.
Yeah, so it's basic solution, it'll be more stable. And then if it's extremely basic, it's going to be basically denatured. And then you can basically reverse that by adding an acid. Actually, I lied. Basic is less stable. Am I clear? Basic less stable. Reverse. Okay, don't be confused.
Wait, what?
Okay, pH. pH. If we go, status of stability. Actually, based off, let's do. This is as simple as possible. let's do this. Not this way. We're gonna say stability. This is more stable, this is less stable. And then we'll say if the pH goes up and it's more basic, it's less stable. Around pH 7, you know, there's stability. If you go a little more acidic, it's even more stable. Then of course, you're gonna have a crash in stability if you acid hydrolyze it.
Oh, so I get it in this case, but does that affect that question we just answered, or is this just like a?
Just basic,
Okay,
just so we know overall that we're clear
Yeah.
on what direction stability is. Acidic, more stable, basic, less stable, extremes, both denatured. Okay. Okay, so we're going down the table.
Yeah, I just have a question about the one that's about X-gal and then the blue product.
Yes, so there's a wording that was a little bit ambiguous in two answers. Everyone clear what question we're talking about? Where two of the answers was all white everywhere. Other one was white and blue everywhere. In the question you saw, I think it said without,
It says continuing the leucine.
no leucine
this is no leucine X-gal, so does that mean no leucine and no X-gal, or no?
and X-gal. So this is how the question was originally worded.
It is X-gal.
So I had email over the weekend. And someone said, I'm confused. It seems like it should be the other answer if there's no leucine and no X-gal. Meaning that all of you are thinking about the question correctly. So that's good, you're thinking about it correctly. I updated the question to say with no leucine and with X-gal. And after I did that, I went through the exam to double check to make sure that there's no ambiguous questions. So FYI, if you run somewhere into anything that's ambiguous, where it could be interpreted in two ways, just apply whatever logic you learned from class. You'll get credit for your answer as long as the logic is correct. Yes.
And then it said with no leucine, but how does leucine, like, is that how that throw off, because leucine just ampicillin and lactate?
Exactly, leucine doesn't matter.
Okay,
So you understand how oxygen works, you know, leucine is just a random nutrient. Yes,
And then the reason why it's built with blue and white is because not all of them take up to the plasma.
exactly. So transformation is never 100% effective or efficient. Okay, I'm glad you brought that up.
Does
Yes,
it say in the question that the plasmid, I'm sorry, I don't have the bullet pronoun, but does it say that the plasmid is not 100% acidic? Because I thought like, you said like, oh, if you're given the right nutrients system that we can. 100%.
no, so as soon as transformation works, if the plasmid is a competent plasmid. And in the questions you'll see, I'll write a replicative competent plasmid, meaning that it has a centromere, origin of replication, all the stuff that needs to work. But a transformation is never 100% efficient, and that's experimentally proven. That's just something to know. That one's just memorize that. It's never 100% efficient. You can't transform everything, or else you wouldn't have to do this whole entire selection process in the first place. Yes,
So, for this question, let's see if I just show you. So this one, I don't understand why my answer would be incorrect, because I thought if we add the protein, you will get rid of the protein, you will have naked DNA. It's gonna break up in like really small fragments. We have like kind of like different really small fragments. It would be like smeared down the gel. But the
so
correct
we'll
answer
come back to
says
that question, and if they're really, really small, where are they gonna end up?
at the bottom, but it says no signal. I
So
don't
if they're,
know.
if they're really small, you actually can't see them. So we'll come back to that. So just hold on to it. We got time. Okay, any questions about one of the no leucine and X-gal? Okay, let's keep going around. Yes, crosslinker. So how this experiment works, we didn't go over in class. It was in your reading, and I also put it on the practice exam to make sure that you're aware of the concept. So remember we said that the histone, when it's not a full octamer, that H2A, H2B are heterodimers. And then H3 and H4 are heterotetramers. So if you think of let's say this is H3, H3, H4, H4, and they're a heterotetramer. When you run it on a gel, what happens to proteins that are not covalently linked or associated is that these four pieces will separate onto the gel. So say, for example, this is the gel. This is just for demonstration. We'll get 4 different bands. So say this is actually two different bands. Say this is H3. And say this is H4. It's just for example. But if we do a process called cross-linking, we actually covalently link these different proteins together. So now some of them get stuck. In the experiments that is shown in your book and the results that you actually see on your practice exam is from cross-linking not being 100% efficient. So you can get these individuals, which would be the H3 and the H4. You could get this whole entire tetramer, which is all four together, which is if you annotate it, that's H3, there's two of them, there's H4, there's two of them. You could get maybe just H3 and H3. So let's say that's H3 and 2, H4 and 2. And then these are all the different bands that you're seeing on that gel. So that's what that experiment is. So say for example, we did H3, H2B, right? Exactly. So you got that down pat. So H2A and H2B only come in dimers. H2A, H2B, they're only in a dimer. And then so if we ran them on a gel and they fell apart, we have the two bands. We have H2A, H2A, and H2B. If we cross-link them, we would end up getting a third band that has a dimer. That'd be the H2A, H2B. So the cross-linking allows these pieces to be covalently stuck together so that when we run it on the gel, it forms a heavier or bigger band, larger molecular weight, higher up on the gel. So is that clear? Yes. Okay, so let's draw it out. So say we want the cross-link so that we get this band. And let's go here and let's go to the extremes, right? Let's say we cross-linked and it was 100% efficient. It worked beautifully. So every single protein we have, all the H2A, H2B, all the heterodimers, they're all stuck together. So when we run it, we only see this band with it all stuck together. But then what's the opposite? We tried to cross-link and it didn't work. It wasn't efficient at all. We would only get these individual pieces because they didn't stick together. Meaning that we get two separate bands corresponding to H2A and H2B, as opposed to anything that's stuck together because of the cross-linking. Yeah. Yeah, so this is, if it was nothing worked. This one up here was the everything worked. If you have some of it working, as how cross-linking normally does occur, it's not 100% efficient, you end up seeing some of it cross-linked, some of it not cross-linked. Okay, does that concept make sense? Okay, good. Please let it make sense. Everybody get hundreds. Okay, next question. Okay. You keep going around the circle. If you're going to skip your question, just say skip and then make sure the next person knows to go ahead and ask.
All right, so I guess, when it came to, the question I asked, what would happen to nucleosome if arginine and lysine residues and histones were replaced with glutamic acid and aspartic acid, so making it more negative?
Yes.
It's like the best answers, because you're making it more negative, and I had looked at the amino acid, I totally forgot about that, because it's acidic, so I think
Mhm.
it's plus, but no, it's minus. So because it's negative and it's negative backbone and whatnot, it's less unstable.
Yes.
That's me, okay, thank you, that's one thing.
Okay, cool. Everyone knows what question we're talking about? Okay, so there's a question where we talked about the histones, and we said that it is important that a lot of the amino acids in the histones are actually they're positively charged. That positive charge is important for interacting with the negatively charged backbone of the DNA. So the question asked, if I took some of these arginine and lysines, I replaced them with aspartic acid or other acidic amino acids, it would actually, what would happen to DNA stability? And the reasoning is that if you take the positive charges, replace them with negative charges, all of a sudden you have negative on negative, so you don't have a strong interaction between the histone and the DNA, meaning it's going to be more unstable. Oh, there's a rock. You want to rock that door? I need to stop turning nouns into verbs. This is not a thing. It's fine. Okay, we'll keep moving this way. Yes. 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 Yes, you can. So whatever way works for you to get and access your notes, if you're like, I'm gonna take my exam on my phone and then put my notes on my laptop or vice versa, that is perfectly fine. We will have LAs walking around, just make sure you're not Googling and chat GPT. But the way you take your notes, whatever's comfortable for you, use that to your advantage. So the idea is that if you don't understand the concepts, and those really won't help you, but if you know the concepts, you can go back quickly, refer to things like, okay, yeah, I got that straight and get the answer right. This is what we do in lab all the time. Like, we don't have everything just at our, I don't know, I guess fingertips memorized, but we know all the core concepts, what we're doing, so we know what to look up very, very quickly. Okay, next question.
Why is, it's like, this gel, and it's saying based on the results of the gel, you put the fluid that which one is the base and then comes on that one. Like, how do you know?
Okay, did anyone understand this question? So the most important thing, oh, did you want to answer? So someone's going to stretching?
Well, didn't you say like on the lower ones, put the caliper, so
Mhm.
that for their densities more about times.
Yes, so I want you to remember for any type of gel, where it's agarose or polyacrylamide, we'll talk about that next lecture. The smaller something is, the faster it runs on the gel and the closer it is to the bottom. So we're looking at the fact that in lane 1, there's a lot more stuff at the bottom of the gel, meaning this has to be small. If it's small, that things got cut up more or fragmented more. That's how we know that it's lane 1. The general principle of how gels work. Okay, cool. Yes.
I just had a quick question about something they asked before. So for the one that like we switched the.
Workspace,
chart
you
to
can squeeze in
negative.
here.
So now I was wondering that one of the options was just on H1 would be bound, so would it not be bound? Or we don't know.
Does it have anything to do with how H1 binds?
I don't think so. I don't know how it bonds. We
Nope.
didn't go over it.
We're gonna go over it. So if you see something that doesn't relate to anything we talked about in class, it's one of those things or answers that are meant to throw you off if you didn't study the concept. Yes,
I don't have anything that's like, sweet, just from the book to not cover them like that.
no. Every concept will have been talked about, would have practiced it, would have been on a worksheet, we'll have seen it somewhere. Yes,
Also with the vet, in the, I know for a lecture, it's up to lecture 5, you have questions, the readings, is that only for this first half of the first floor, or are you gonna be more questions that, you know, the reading guide questions that we.
oh, there'll be more.
Okay.
Yeah, they'll get posted probably tonight. Yes,
So then I have another question. My question was about this one about MVT plus that we replaced it with CAT plus. So then, the correct answer is DNA extension will be inhibited, but I don't understand why wouldn't it just be slower? Like, why wouldn't it, instead of like completely being inhibited, I don't understand why wouldn't it just like work slower?
so slower is inhibited. So think of inhibition as like slowing something down. You don't necessarily have to completely stop something to inhibit it. And this is basically like lab jargon or lab lingo, when we say inhibition, is reducing, putting on the brakes.
Um,
So yeah, it would slow down, it would inhibit it.
But like, so you're not, when you say DNA extension will be inhibited, you don't mean like DNA extension will just not happen, like it wouldn't go forward?
Correct.
Oh.
Okay, where are we in this circle? Okay, how about these 4 people? Floor person? I was sorry. Okay, we're here at the end of this middle circle. Any questions that you have?
Another one was, one of the there was like a question that was select all that apply for CRISPR,
Mhm.
and then like it doesn't have pyrophosphate or whatever, like it just doesn't work. I'm not understand, like one of the options that I missed was they have an accumulation of toxic pyrophosphate. I was wondering, is the toxic part anywhere that I missed?
So we didn't go over the fact that accumulation of pyrophosphate is toxic. It's toxic because if it accumulates and you're not having DNA replication. So don't worry about that topic in the exam necessarily, but I do want you to know that cells cannot amplify their DNA if they don't have pyrophosphatase. So that's the take home point.
So they want DNA will stop.
Yes, especially,
That's a question.
exactly.
And then.
Okay, go ahead. Oh, okay. The question that you brought up earlier, it was, okay. So we're gonna draw this one. This one is our experiment where essentially we had the repeating units of 200 base pairs of DNA. 200, 400, 600, 800. And then it asked if we. If we first treated with proteinase followed by a lot of nuclease. So let's do that. When you add proteinase and you do this first, what do you think happens to your sample?
Yeah, rid of the nucleus.
Okay,
So
so
I just didn't make it.
because those are gone, we've got naked DNA. And then I said we add a lot of nuclease.
Yeah.
This a lot matters. What do you think is gonna happen if we add a lot of nuclease? Little tiny fragments. If we run really tiny fragments on a gel, where do they end up? Very, very, very bottom. Have any of you ever run an agarose gel in the laboratory? If they're at the very, very bottom, they're really, really tiny, even if we try to visualize, can we see them? We can't see them because they're so small. It doesn't emit enough light from whatever's circulating or binding to it. So you usually get either some signal at the bottom or no signal at all.
But I was like thinking, like how it was similar to the lane 1, lane 2, lane. So we wouldn't get something like this, like lane 1, like it's all mostly at the bottom, but.
Yeah, but these are very, very, very tiny pieces. And then so what you could think about is if you had some nuclease, or you did it for a short amount of time, you could probably generate a smear. And that would give you a smear so you can get some bigger pieces along with smaller pieces. Because in that one, where there's like lane 1, lane 2, lane 3, lane 4, you notice there's really big pieces there too. But if we're not going to get a smear and we're using a lot of nuclease, when we cut it up, it's going to be very, very tiny.
And also the protein since there's no signal or DNA at the bottom of the gel. I thought it means no signal and there wouldn't be any.
Said or.
So
either all your signals at the bottom, depending on how big it is, because it's small, or it's so small that you can't get any signal.
Okay.
Yes.
Could you repeat what you said about adding proteinase?
Okay. So we said, what if we add the proteinase first? We cut up all the histones. Remember, the histones is what's protecting the DNA from getting cut up in this experiment. So now if we get rid of all the histones, we only have a whole bunch of naked DNA, meaning that everything is susceptible to getting cut up by that nuclease.
So the like fragment size, like typically would change or just.
Yeah, it gets more fragmented. And the reason why we have these repeating units is because first we had the protection of the histones about every 200, right? That's really how this whole experiment came about. Okay, 2 questions. Let's go here.
This is just like a quick and follow-up question. So the reason that it would all be at the bottom, like the beginning, is it because it would be really fragmented?
Yes.
So why it's gonna be all, okay, got it. I just had a follow-up question on this. What if like we were mixing added a lot of nuclease and then it's what it changed? How the gel.
What do you think? Let's walk through it.
We'll just be all at 200.
I think that was actually based on that question.
So we said if we do, so the original experiment is some nuclease or a small amount of nuclease. That's the original experiment where we got these 200 repeating units. And you asked if we did a lot, which we put on this side so this is a lot of nuclease. So if you have a histone, you have DNA wrapped around it, and you add a lot of nucleus, what do you think is going to get cut off? And the only part that gets protected is going to be what? On the histone. So then if we add proteinase, we get rid of the histone, now we have a little bit of DNA left. How long do you think this DNA is? 150. So remember what wraps around the histone is the 150. And if you ran it, you'd see 150, basically only 150, because you're not going to have pieces that are still stuck together, represented in the nucleosomes.
So all of them are just.
Be this 150. So you
What happens to the liquid? Wouldn't we
it got chopped up like really, really badly by this a lot of nucleus.
So the protein wouldn't affect the DNA on the histone?
Correct.
But there was a scenario that we would only get the 200 base pair. What was that?
So 200 base pair is the original experiment. So let's draw this.
The one that we only get 200, not 204 and 6. I
Where-
remember there was a question that we would only get, like, it was only 200 base pairs. It's not a.
I think the question was, what does the 200 band represent?
So there's no scenario where we all, like it cuts efficiently to the point that all of the fragments will be 200, 200, 200, 200.
No, there's not really a way to cut efficiently and also protect all the linker DNA. can't do both. And the reason why this whole experiment worked out, because you're able to have, say, like one histone with linker DNA, and then 2 histones with linker DNA because of the inefficiency, 3 histones with linker DNA. Okay, we just had a third question, same topic?
Oh, yes, I was just wondering, the nucleus is added, in the original experiment, the nucleus is added first to the. Or
Yes, so the nucleus is added first so that you get cuts.
just a little bit.
Yeah, just a little bit's added first. And majority of the time, so what happens is that this whole set of like histones and DNA, you're cutting where it's most accessible, which is in between the linker DNA. Sometimes you cut it off and you only have 1 piece. Once you cut it, there's 2 pieces. Once you cut it and there's 3 pieces stuck together. And that's why we got these repeating units of 200.
In the question I asked you was, it says you add the proteins first,
Mhm.
so it is able to capture or get into the nucleus.
Yes. Even the DNA's there, protein can still degrade histones, even though the DNA's on it. So the protein protects the DNA does not protect the protein.
Okay, so then the protein is too forgetting.
Yes.
Okay, so that's not the nucleus. Thank you.
Oh, no, so the protein still gets got. Okay, any more questions on this topic? Yes. Okay, we're still starting, okay, we're down here somewhere with questions then. Yes,
So one year when we talked about this, so we kind of asking like what were the DNA look like? You were referring to the one that was on.
yes,
You weren't asking the hypothetical that UNC 400.
no, I wasn't asking that. So basically, what do these bands represent? This is what these bands represent. Okay, any other questions on this topic? Yes, you would go with proteinase first, get rid of protein, and then a moderate amount of nucleus. So you can just kind of get cuts in all different types of lanes coming out. Yes,
So how come some of the original experiment, they still had some of the nucleus?
because they use a small amount of nucleus. It's all relative.
So they use a bit of nucleus, yes, they don't.
Yeah, use a whole bunch of DNA, you're gonna cut whatever's like hanging on. Okay, yes,
You say for, you would use also near the proteins that are moderate in that nucleus?
mhm. Okay, anything else? Oh, yes,
So then the small size is.
yes, exactly. So as long as they're on histones, they're protected, they're not gonna get cut up. Okay, any other questions on this topic? Okay, we'll keep going. We're gonna do floor pod. Any questions on this quadrant?
I just had a question on the reading guide. I saw there was a question asking about like the engineering experiment. I was wondering what's that.
Okay, so interest of time, when I set up the course, you'll see that the reading is broad and then we focus. That's when it comes to examination, I'm testing the core concepts. The reading guide helps you build the, what do we call this? The framework for understanding actual concepts that we're going to go over, but when we test, we're not testing on them. So when you go to study, don't take the reading guide and try to help yourself study. It's not gonna help you. Don't go back over the reading over and over again. It's also not gonna help you very much. Don't take your time going over all the reading quizzes. That's also not gonna help you. Spend time on the homeworks and the practice exams and the worksheets from discussion. That's what will prepare you for the exam. So we can come back to it at the end of the exam. Yes.
Extra questions other than the practice exam? Or if we
Practice
redo that,
exams, the
is it-
homeworks, and the worksheet.
And if we redo the practice exam, are there like new questions in it or?
No, yes.
My question was.
Question 10. Ooh, Messelson install. Okay, so we have a Messelson install question. Basically, we're repeating the experiment. So let's go backwards. Messelson install is normally 15N, and then you move your cells to 14N. And then I asked, I'll do something fancy. And then you ask, I repeat the experiment, and instead I swap my growth media. So I'm starting off in 14N, switching it to 15N. And then it says I incubate my cells, 2 generations, so it's going to be 2 generations. And then we run our results on alkaline gel. Alkaline. Okay, so we got the question stem. So first thing I need you to understand, which one is the parent strand, light or heavy? Light. Light is going to be your parent strand because that's where we grew it first. All your daughter strands are going to be heavy. So now the next thing is that we grew it for 2 generations, and I always like to do this with markers. So for first generation or parent generation, you have your DNA strand. Your second generation, I have a parent strand, they both separated, and then they added on 2 heavy strands for the daughter strands. So that's generation number 2. I mean, generation number 1. You go 1 more generation. Go here, here. And then here. They separated, added on, no. So generation 2. So now we're at exactly where the question told us to be. So the last thing is that it told us we ran this on an alkaline gel, meaning that we're going to separate all these into their initial bands because this is denaturing conditions. So if I take my pink marker, put these over here, I got 2 pink bands. I have 4, 5, 6, 1, 2, 3, 4, 5, 6, 6 green bands. Parent, daughter. It's asking me what type of bands do I have? I have 2 types. I have light and I have heavy. So I end up seeing a light band and a heavy band. So does it make sense how I worked through that question? Yes. Yes. So normally the original experiment, the parent strand is heavy. The original experiment. Original. The question stem stated that, oops, I switched my growth media and I grew it first in 14, which is light, and switched it to heavy.
So the roles reverse.
So the roles are reversed. So just be aware of that. At the end of the day, for this particular question, it didn't matter because it was an alkaline gel. Light, heavy, wouldn't have made a difference. But say this was a, what do you call it? A native gel, then the answer would have been different, depending on which one was the parent.
Right.
Mhm. Okay,
So the reason that we don't have a mix is because it's an alkaline.
exactly. So alkaline separates those strands.
So the only one that gives you mixed throughout is native, right?
Yeah,
Or
it's native or if like it's dispersive replication. If it's dispersive, it's always mixed, right? It's mixed everywhere.
even in alkaline.
Mhm. Yeah, I see that. Okay. Any more in this quadrant for questions?
So in this question, we want to say, something about it's transforming into a blue state. But I was just wondering like, what that means, like, would it be guaranteed that way soon, or it has the chance to get that?
Oh, so transformation and efficiency.
I think so.
Okay, so we did that right at the very
The
beginning.
question where it has 2 answers.
Yes. Okay, so I just want to reiterate that for transformation, remember, it's never 100% efficient. Here, I'm gonna take the question. Okay, so this is the one that we did in class. So we've said, basically a leucine oxytroph was transformed with LU2 containing selection marker.
Yeah, what does that mean?
So transform means that the DNA gets put into that cell type.
So it's guaranteed that cell type,
Or if we take that DNA, we put it into that cell type. That's what transform means.
but would it be that leucine? Or
So it picks up the gene to synthesize leucine. So the transformation is the transfer of, this is genetic material. This is a cell. The transformation of this genetic material into that cell. That's transformation. That's pretty. Okay, that's transformation. Genetic material into another cell. Yeah. Okay, so repeat. We have. Sure, okay, good. We have a LEU to delta, this is delta not A, delta cell. What does this mean? LEU to delta. Yes, so this is a leucine oxytroph. And then we're going to transform, what does that mean? We're going to transform LEU2. What is LEU2? Anybody? Yes, that's,
Leucine producing.
yes, this is the gene for the cell to make leucine. So now my cell, which didn't have leucine, is going to get this LEU2 gene, which means I now have a cell that has LEU2 and that it can make its own leucine, and it's not going to die if it doesn't have leucine. And then so it asks, what happens if a cell doesn't get the plasmid? What happens to the cells that don't get the LEU2 gene? They won't be able to make leucine. If they can't make leucine, they'll die if you don't give it leucine. Is that clear?
Oh, it just, I didn't understand. How the plasmid would have gotten that gene.
So it's
But it's
because,
just for the other plasmids, I guess, or?
so it's just for other cells. So think of every transformation as a whole bunch of cells. You know, lots and lots of cells. When you take this gene and you're trying to put it into the cells, not everybody gets it. And it could be anywhere from 30% get it, to 50% get it, to 80% get it, but not everyone gets it. So you're always going to have a mix of transformed cells and what you started out with. And the question is asking, what happens to the cells that you started out with that didn't get the gene?
And if it was the cell that didn't get the gene, you know, they don't really need it.
Exactly. Yes. Okay, so now we're here.
Well, my question's right.
Okay, keep moving down.
One of the questions is that, we can check the bacteria and discover the very different that confers ampicillin and biodiversity.
Mhm.
So this simulation is converting a R.
Yes, so confers means it gave them the ability to become resistant. Once again, like scientific jargon, you'll see that kind of in different courses you'll take. Okay, we'll keep going. Okay. Okay, oh, it's fast. Okay, there we go.
So on that, question we just talked about, with the phase. I forgot, the resistance. The way I understood it is that it would stay by when you really so I know it's
Yes,
that was difficult after they were trying.
okay, can you read me the whole question,
Yeah.
Stanley? Make sure we're all clear.
You isolated an emerging understanding of bacteria from infected patients using infected bacterial genes. And discovered that we played that antigen that confers 10% antibiotic resistance. Those are the following best defenses in their type of the. Resistant. Okay, oh, okay. Okay, yeah, it's an episode. So it would die.
It's dying.
Okay, so does everyone see that question or know what question I'm talking about? I want to make sure everyone's clear. Is anyone still, yes? Okay. Does anyone have that question pulled up so I can read it out? Okay, I got one. Okay, 21, 21. Okay, you isolated an emerging ampicillin-resistant bacteria, Pseudomonas, from septic patients. You genotyped the bacterial genes, discovered that they carry an AMP gene that confers ampicillin antibiotic resistance, which is the following best describes the phenotype of an AMP delta mutant Pseudomonas strain. So the actual question, if you think of the first two sentences, the only thing you get from there is that ampicillin, what the gene does. That's what it does for you. The actual question is, what is the phenotype of the AMP delta mutant? So know what that delta means, and basically if you don't have resistance, you'll die in the presence of ampicillin. So take your time to read the question thoroughly and ask yourself, what is this question asking you? It can be easy to go into autopilot, especially if you're familiar with the concept and get through like the first two sentences and think you know what the question is asking. Make sure you know what the question is asking. Okay, that was good. That was great.
Yeah, thank you.
Welcome. We're over here. I'm not shooting y'all. I'm gonna do different fingers. Okay. I don't know what fingers to do. Okay. We're here. Anyone in this quadrant? Okay, I'm back. So no discussion Monday and Tuesday. If you're looking for a discussion schedule, double check the syllabus. I'll make an announcement because apparently it was not clear, my bad. So there's only 6 discussions total for all 10 weeks. Next round of discussion starts Wednesday, it's Wednesday through Tuesday, a week off from discussion, then a week of discussion, and then Wednesday through Tuesday. That's how it's set up. Yes,
Can we, go over the homework too now? or-
yes, we can get homework too.
Also, one thing. So
Yes.
for the fluorescence, are we supposed to know that?
Yeah, so in lecture, I talked about what fluorescence was. So this was the basic definition. Fluorescence is when you have a molecule, it gets hit by one wavelength of light, it releases a different wavelength of light. Yes.
This is one word question on the top, because we have at least one that was asking like why it's like calcium two plus two work is an energy plus for the DNA family. And the answer is that it's winning, and that became dollars.
It's inhibited. So it's a special property of actually magnesium in that cation. And then so you just have to know that magnesium is a cation that DNA polymerase needs in order to extend. There's only one exception in nature, at least that I know of, that uses calcium, and it's a very, very bad polymerase. It does a sucky job. So basically, if you try to replace magnesium with calcium, you basically just do a really sucky job and you inhibit DNA synthesis. Yes.
The question where it asked about the, like, the COVID -19 span, I was a
Mhm.
little confused by that. Can you explain why it'll form hybrids of DNA? Like, how do we know that we have a DNA strand?
Okay, so the concept in that question is about using a viricell to basically try to figure out what type of virus COVID is. At the end of the day, what we want you to know is that RNA can hybridize to DNA. So it doesn't matter if it was an RNA virus or a DNA virus. If you have a probe that had like similarity to it in that viricell or in that virichip, then it would hybridize with it. So just knowing that RNA can interact with DNA and hybridize. Yes.
On the question with the curve of DNA, what is the x-axis? Is it?
What happened?
Your question was what is the x-axis?
Yes.
Axis on the curve.
Yes.
If the x-axis was pH, what would it look like?
Okay, let's write it out. Dude, this pen sucks. Where's my paper?
Yeah, it doesn't even sound really.
Are you serious? The green doesn't show on video? It's like a filter. Okay, keep going me. I got blue.
Oh, yeah.
Ha 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 ha. Okay. So this is gonna be absorbance at A260 nanometers. We're trying to figure out what would different x-axes look like. So the thing we need to understand and remember about this absorbance is what causes absorbance to go up and down for DNA.
The knowledge, it's double stranded,
Okay, so if it's double-stranded DNA,
it's down.
what else?
Single stranded, it's-
Yeah, single-stranded, going to be higher. And basically, this is like the hypochromic and hyperchromic effect, right? The more stable and condensed the DNA is, the less light it's going to absorb, and the more loose it is, and open it is, the more light it's going to absorb at 260. So if we know something about pH, right? And say this is, we'll start at pH 7. What do we expect the DNA to be like at pH 7?
Low, low, single stranded,
Low absorbance because it's stable. If we go acidic, where do we expect this to go?
slightly goes down, small.
Maybe a little more stable. What do we expect to happen if we go extremely acidic with denatures off of fast? Okay, what about with basic? If we go basic, what do we expect?
No,
A little unstable. You start increasing this. If you keep going more basic, what do you think happens? We're starting to denature. So if temperature, you see where as you get colder, it's stable, 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 nothing else happens. pH, you can see that there's a U-shaped kind of relationship where there's instability at both ends. And then so the absorbance would increase. Is that clear?
What would be any other example? I mean, we are not going to ask about strong.
Nope. Doesn't matter. Does DNA concentration matter?
I mean, I think it's about one.
If you have more DNA, is there gonna be more absorbance?
Yeah.
Mhm.
So they just, how it works. is just-
Okay, you're right on track. So let's say this is DNA concentration. What do you think the relationship is gonna be?
I'm sure how, oh, linear.
Mostly, yep. More DNA, more absorbance, right?
So it wouldn't be exponential.
No. I mean, you get to a point where like the signal gets saturated. But for the most part, it's a linear relationship between absorbance and the knowledge.
So these. But these
Not substantially, no. Any other questions about this concept?
Is there anything else?
Nope, nothing I want to ask you about. Okay, so we are at 8:50. The plan is to move to HLSH because we should have more space. There's a whiteboard in that room, right?
Yeah.
Okay, we're gonna move over to HLSH 200. So we'll go there together and then we'll continue. I'm gonna stop this recording. So it'll be part one. Then we'll pick up off with homework.